Welcome back to the course on fluorescence spectroscopy from the CFS. Today is part 5, and we're going to be talking about fluorescent probes. This is a vast area of research, and it's hard to talk about, and it's hard to organize because of the wide number of probes that have been used in a wide number of applications. It's really talking about probes is like trying to describe biology in one or two lectures. It just can't be done. So in part A today, we're going to focus on the most commonly used probes that are completely organic, and we'll leave the inorganic probes to later time. And we're going to focus on classes of probes to try to make it sound more coherent. And what has happened really is when fluorescence was being used as a tool in biophysics, we had a relatively limited number of probes, and that was enough. But it was the applications to cell biology that had really resulted in expansions of the probe library. So what are we going to talk about today? We're going to talk about intrinsic probes, extrinsic probes as general markers. Many of these have a second generation version that are improved. And we'll talk about ion sensing probes and protein sensors and provide examples of each class of probes to show how they are used. I showed you this slide in the first lecture that there are basically two types of probes that are natural to biology. They occur by themselves. These are the intrinsic probes. And then there are extrinsic probes, which are something we add to provide a signal that otherwise wouldn't be there. So just for the moment, we're going to talk a little bit about intrinsic probes. And the most obvious intrinsic probes are the aromatic amino acids that occur in proteins. In proteins, there are 3 aromatic amino acids: tryptophan, tyrosine, and phenylalanine. As you can see, the tryptophan is the longest emission wavelength of the probes, and it turns out to be the dominant emission that's observed from almost any protein. So, tryptophan is present in a relatively small percentage, 1.1 mol percent in proteins. It's the least abundant of all the amino acids, and that's probably good because more tryptophans just result in a more confusing signal. And most proteins contain more than one tryptophan. I'd like to draw your attention to the line here at 300 nanometers. This shows that tryptophan can be excited almost exclusively at 300 nanometers. So if you wish to avoid any contributions of the other aromatic amino acids, you can choose something from 295 to 300. Even if tyrosines are present in the protein, you normally, which you might see if you excite around 280, the tyrosine maximum, you usually won't see the tyrosine emission. A lot of it is transferred by fluorescence, resonance energy, transferred to the tryptophans. And phenylalanine emission, I had to search far and wide to find an example of that. So it's never observed unless your protein or peptide does not have any tryptophan and does not have any tyrosine. We are fortunate that tryptophan is so highly sensitive to its local environment. So as an example of that sensitivity, I've chosen this single amino, this protein azurin that has a single tryptophan residue, and in the native state, the tryptophan residue is shifted very much to the blue to short wavelengths, indicating that it essentially has no contact with water. If you denature the protein, the tryptophan shifts to much longer wavelengths, so that gives you the tryptophan emission is always going to be sensitive to the contact with water. Exciting at short wavelengths, you'll only see the tryptophan. In this particular case, if you excite at the shorter wavelength, you can see some of the tyrosine emission on the edge of the tryptophan, but that's sort of unusual to obtain that result. Oh, and we're going to have complete lectures on protein fluorescence. That's why I'm not going to go too much into it. As far as the intrinsic probes, we have NADH, which is the one that is nicely fluorescent and is widely used. And we have flavonoadenine dinucleotide, which has very weak emission whenever it's bound to proteins. It's good in solution, but not when bound to proteins. So the emphasis in research has always been on using NADH for some type of biophysical experiment. So how does NADH behave in solution? Well, it's a pretty large flexible molecule, and it's quenched by collisions with the adenine. So the NADH fluorescence is not as bright as it could have been because it's always bouncing against the adenine. And so, but protein-bound NADH is almost always in the open conformation, keeping the adenine away from the NADH part, and as a result, the NADH emission increases on protein binding. And you can see this in this experiment where we have NADH binding to a steroid dehydrogenase. And if I draw your attention to the lower panel here, we have the emission of NADH free in solution. We have the NADH when it's bound to the dehydrogenase. Its intensity has increased about fourfold, and the lifetimes are quite different. The lifetimes of free in solution of NADH are around 0.37 nanoseconds. But if you bind it to protein, it goes up to 1.2 nanoseconds or so. So the answer for such an experiment is pretty clear. If you're exciting at 340, where you're only exciting the tryptophan, the NADH. If you excite at 280 nanometers, well, in this case 295, you see both the protein emission and the NADH emission. Protein emission is decreased a little bit because of threat to the NADH, and just like in the bottom slide, the NADH emission increases about fourfold when it binds to the dehydrogenase. These changes have been very useful for detailed studies of binding of NADH to proteins. So I'd like to explain this experiment to you because it's illustrative of what's been observed. Let's take NADH first, just by itself in solution, and the increase in intensity with NADH concentration is linear. If you have protein in the solution, you find that it's not linear. At first, there's a much faster rise in the NADH fluorescence, and then it levels off. What is happening here is these are the NADH molecules that are binding to the dehydrogenase. Then the dehydrogenase gets saturated, can't bind any more NADH, and you go back to the linear curve where you're just looking at more free NADH in solution. Okay, now we're going to move on to classes of extrinsic probes. I'd say that there's probably more than 3 classes, but I'll divide them up at the beginning into 3 classes. First, we have the fluoresceins and the rhodamines, which have been the most widely used probes. Then we have the alexafluors, which I would classify as a second generation probe because they're an improvement on the first generation fluoresceins and rhodamines. And the Bodipis are another second generation probe that have some advantages and then also some disadvantages. So we'll discuss each of these in turn. Fluorescein was used very early as a probe. Actually in 1877, there was a question whether the two rivers in Germany, the Rhine and the Danube, were connected to one another. And the way that was determined is a scientist decided to put a bunch of fluorescein into the Danube, and actually he put like 10 kilograms in. He put an enormous amount. And some 60 hours later, they found fluorescein showing up in the Rhine. So that proved that these two rivers were connected in some way by an underground connection. That might not seem like a relevant experiment, but even today, fluorescein is widely used for mapping flows of fluid, in this case, blood flow. How do you do a retinal angiography where you want to image the blood vessels in the back of the eye? And that's done by getting a large injection of fluorescein and taking a picture of the back of the eye. And wherever the fluorescein is, you can see the veins or the arteries. And this is used for the diagnosis of macular degeneration, because if there is macular degeneration, you see the macula increasing in size as observed by fluorescein due to leakage of blood out of that area. This was fun. I actually wanted to, this is not my back of my eye, but I wanted to see how this worked one time, and somehow I got the hospital to do that for me, so I got my own eye imaged. Also imaged with a red dye that sees a little deeper into the eye, but I won't confuse you with that. I would say fluoresceins and rhodamines are the most widely used FRET pairs. As you saw in an earlier slide, they're available over a range of wavelengths, or at least the rhodamines are. There's not as many fluoresceins. And you can always find a pair with good spectral overlap. So you can do a FRET experiment where the fluorescein is excited and is transferred to the rhodamine acceptor. And here's an experiment of using fluorescein and rhodamine for proximity measurements. This is an assay for the HIV protease cleavage enzyme. This is important because in order for HIV to multiply, there have to be multiple cleavage sites for the replication, and that's what the cleavage protein does, and it cleaves a particular sequence. So one way to assay for this protein is to make the appropriate sequence, add the appropriate donor acceptor, and if there's no cleavage protein in the sample, the energy transfer won't change. But if there is the protease protein, this peptide will be cleaved, the donor and the acceptor will diffuse away from one another, and the energy transfer would go away. This is a bit of a complicated example, but I wanted to use it because in my lab for the past years, we've been focusing on the near field effects on fluorophores near metal surfaces, which has turned out to be rather interesting. And I found a FRET experiment on an aluminum surface, Actually, it's a aluminum nanopore surface in which fluorescein and rhodamine were put into it. And we have the emission spectra here. Here's the emission spectra of the fluorescein alone, and the emission spectra of the fluorescein after the rhodamine is put in. The dashed lines are the separate spectra for each species, but the important thing is that the fluorescein is greatly quenched. The rhodamine is greatly enhanced because of the energy transfer. And there was probably more energy transfer here than could be expected because we have observed an increase in parent Foster distances for fluorophores near metal surfaces. So I wanted to use well, this is not our experiment, but we have been working on such samples. One of the largest problems with fluorophores is their photostability. And something I meant to mention in the first slide, when you first open the catalogs or start looking on the website for fluorescent probes, you could easily be overwhelmed because there are so many different probes and so many different ways they are conjugated to proteins or lipids. It's just mind-boggling. And you want to choose something that would be the right probe for your experiments. So, for instance, if you were planning to do some type of fluorescence microscopy experiment, and if you would choose fluorescein, you were doomed because fluorescein photobleaches very rapidly in the high intensities that are used in fluorescence microscopy, and you can just watch it disappear before your eyes. So you would want to choose something that is more stable. And this has led to some of these next generations of fluorophores where the alexafluors and the bodipes would show less rapid photobleaching. So let's look at the alexafluors. These were the next generation of probes that came out. And just like the fluoresceins and rhodamines, they're available over a wide range of wavelengths. I would call these the second generation probes. And as you would be expected, the longer wavelength probes are larger, have a larger aromatic structure than the short wavelength probes. I didn't realize till it was time to prepare for this lecture. I always wondered, why they made these Alexa probes. Well, they made it to make them more stable, and they made them because they have slightly more narrow spectra than fluoresceins and rhodamines, so you can fit more probes into a single experiment. But I never knew why, and here's the example of the being less prone to fluorescence quenching. So fluorescein is bleached very rapidly. The alexafluor is not. And another favorable feature of the probes is you can label your macromolecules more heavily with an alexafluor probe than you can with a rhodamine probe, which Texas Red is. That's an advantage because in many experiments, brightness is important. But why did these probes work? And I found out surprisingly that they are basically the same probes as the fluoresceins and the rhodamines, except sulfonic acid residues have been added to these probes. And they gave them a different name, but now I know how they're made. And I really think this was pure genius. I've been looking at fluorescence probes for a long time. I personally did not realize that the sulfonic acid residues could impose increased photostability to the probes. And another thing, it allows the proteins to be labeled to a higher degree. And I would have to guess that's because the negative charges forces the probes when they're binding to the protein to bind further away from each other. This binding is often covalent. I haven't listed all the different ways that probes can be bounded to proteins, but there's the N-hydroxy-6-cinamides. I guess that's the only ones that I have here. There's a whole range of different, when you buy these probes, you can buy them either in a reactive form or a non-reactive form. The reactive forms might be reactive with amines or carboxyl groups or sulfhydryl groups. This is always on the surface of the proteins because they're so highly water soluble. They're not going to penetrate the interior of the protein. So that's another thing you have to consider when you're buying the probes. Do you want a reactive form that is going to automatically bind to your, covalently bind to your macromolecule, or do you want to buy a non-reactive form? The next set of generation probes are the BODIPEs, and these are interesting, and they seem to have more potential than the other probes because the chemistry, well, maybe the chemistry isn't easy, but I've seen so many different elexifluors made sensitive to different ions that I think their use will continue to grow. Their emission spectra are more narrow than either the fluoresceins, rhodamines, or the elexifluors, and this means you can fit more experiments, especially in something like flow cytometry, where you'll want to measure different molecular markers on the surfaces of cells, and so you'll want to use different antibodies labeled with different probes, so you can squeeze out more measurements if you use probes with small emission, with narrow emission spectra, and these are usually not very sensitive to solvent polarity. But they do have one major disadvantage, which is they have a very small Stokes shift, which means they have a lot of overlap of the absorption with the emission spectra, and this results in self-quenching. The experiment one of the experiments that shows this is the binding of this biotin moiety to the avidin protein, which has 4 binding sites. They're distant from one another. And if you add different amounts of the avidin to the BODIPE solution, you see the fluorescence going away as the probe is binding to the protein. So that's the self-quenching that occurs, and it must be a through space interaction, at least in this case, because the BODIPE groups are held far apart by the distant binding sites on the avidin molecule. And not only has avidin been used for just plain labeling, many biotin probes have been made for many ionic species, and I just listed one here. One is rather an unusual one. It's a probe for mercury. You have the biotin moiety here, the chelator for mercury here, and when you put these into cells, you find that without mercury there's no signal. With mercury there's a signal. With copper there's no signal. So this particular probe is highly specific for mercury. I personally wouldn't think there would be a reason to measure mercury in cells because there's going to be so little in any particular cell, but perhaps I'm wrong about that. This is something that is general for all of the probes. Just about all of the probes are available in two forms, a membrane permeant form and a membrane impermeant form. And what the difference is, the impermeant form has the typical ionizable groups are hydrolyzed. The permeant ones are the acetoxymethyl esters shown here. So they're nonpolar. They go across the cell membrane, and when they go into the cell membrane, they are hydrolyzed by esterases in cells. And actually, esterases are pretty chemically stable. It's hard to hydrolyze an esterase if you have one. I've looked up methods to do it, and it's pretty difficult. But this happens to be a calcium-sensitive acetoxymethyl ester probe that is sensitive to calcium. So this diffuses spontaneously into cells, it gets hydrolyzed, and it gets trapped. And an example of its application is doing a neuron where you put in the acetoxymethyl ester, it gets trapped, and then it's seen throughout the neuron as needed. And I don't know why that other dextrin labeled rhodamine is in the picture, but the important thing is they're membrane permeant, and you can wash away anything that's outside the cell. Here is a trap that is easy to fall into. And you have a certain instrument. It might have a laser light source that is at a particular wavelength, and you put your sample in and you find no signal. And one reason that can happen is because all of these probes, the fluoresceins, the rhodamines, the Alexas, they all have relatively sharp absorption spectra, and they all have areas, wavelength areas, where they display very low extinction coefficients or very, very low fluorescence. So if you use one of these probes and your instrument is exciting, say at 400 nanometers for BODIPI, you're not going to see anything. So you have to be, you can be fooled by that. So I wanted to mention that these probes all have a blank area where they don't absorb much light. I might have the physics all wrong with this, but I found this to be an interesting thought as I was preparing this lecture. I was thinking to myself, what is the size of the photons and what are the sizes of the fluorophores? And in a way, it makes absorption a remarkable phenomena, because according to quantum mechanics, a photon is about the length of the wavelength, so they're relatively big. These wavelengths are bigger than any protein, and they're much bigger than a fluorophore, which is really more than a nanometer or 10 angstroms in size. And it seems remarkable to me that these 1 nanometer size fluorophores can absorb 400 nanometer photons. So just think about this for a minute, and I'll do that by showing you a list of Alexa fluors and a list of extinction coefficients. Remember, the optical density of your sample is given by the product of the extinction coefficient, the concentration of the fluorophore, and the path length. And these are the extinction coefficients in reciprocal mole or reciprocal centimeter units. And you can see they can range, over a factor of well over a factor of 10. And what does this mean in terms of this getting this large photon to interact with the small fluorophore? So I put this figure together because I knew something that I've read in many places, that if you, instead of using the extinction coefficient, you calculate what is the cross section for absorption of light by a fluorophore, and it turns out that the cross section of light is always about the size of the fluorophore or much smaller than the fluorophore. So if you have something which it has well, I'll start down here, where you have a high extinction coefficient, a larger fraction of the light will be absorbed and a smaller fraction will pass through. If you have a molecule with a low extinction coefficient, only a smaller absorption will be absorbed and fewer will pass through. And so in the case of absorption, it seems to me like photons act more like particles than waves, because it's the physical cross-section of the fluorophore that it interacts with the photon and not the cloud of energy around the photon. All right, let's switch to other types of labeling of macromolecules. One method is designing the probes to mimic macromolecules. This is always a sort of an easy way to do it if you're doing any type of membrane experiment, because so many of these are commercially available, and you can choose a phospholipid that has probes way out at the end of the polar part of the molecule, or probes at the non-polar part of the molecule. And many experiments have shown that the probes localize as you would expect from where they're attached to the alkyl side chain. So localized labeling of membranes is relatively straightforward. Cyanin dyes. I should have put cyanin dyes in with the first generation of fluorophores, but it just made things too complicated. These were used very early on for two types of experiments, for FRET experiments and for single molecule detection. Now, the fact that they're being used for single molecule detection tells you right there that they're photostable to a high degree, because you can see just one molecule, and in order to see one molecule, you have to be cycling it very fast, very high intensity illumination. So I'll show you an example experiment where cyanin dyes were used. And this is an experiment which was done with a holiday junction in DNA, and they labeled the different strands with either CY3 or CY5, and the holiday junctions were thought to undergo structural fluctuations, which would make the cyanins and the two cyanins far apart, or further apart, or then bring them closer together. And this was looked at by single molecule observation. And when you're doing that, you're usually not doing spectroscopy, but you're choosing a single wavelength to look at the light from each fluorophore. And you see that the intensity of the fluorescein and the rhodamine, CY3 and CY5, jump up and down. So the amount of FRET jumps up and down. So you can even calculate the equilibrium constant of each form from these fluctuations. But I guess an important point is, even for the most photostable molecules, they all disappear after a few seconds. They can only stand so much illumination, and then they die and the signal goes away. DNA probes. DNA probes are incredibly important, of course, for not only for sequencing, but for doing any type of gel experiment. And there's a couple of classes of probes for DNA. Some probes, like ethidium bromide, intercalate into the DNA double helix, and the ethidium homodimer intercalates and binds even more strongly into double helical DNA. There are other probes that bind to the minor groove of DNA. And the difference of these probes is, well, especially let's take lithium bromide. If you're staining a gel with lithium bromide and you're looking for DNA, you're just about always going to have a background of lithium bromide fluorescence throughout the gel, because you can't wash it all out, because the lithium bromide is going to wash out of the DNA. So the lithium bromide increases in fluorescence when it binds to the DNA, but it also can be diffused out of the DNA. So this resulted in the development of the bisintercalating dyes that started with the ethidium homodimer and then moved on to this next class of dyes, which I think are remarkable, the Toto and the Yo-Yo series. I don't think it's an accident that they were labeled Toto and Yo-Yo, because I think it's a tribute to the old movie with Judy Garland, where her dog had this. I think the dog was named Toto. But these dyes are wonderful for doing many types of DNA experiments, because they bind very strongly by two mechanisms. They bind by intercalation, and the binding by intercalation gives a rigid environment for the fluorophore, so it becomes brighter when it's bound by intercalation. And then it has a positively charged linker between them, which binds to the phosphates. And these dyes just never come off of a DNA once they're bound. So they can be used for things like following PCR, because as soon as the double helical DNA appears in a PCR experiment, you'll see an increase in fluorescence due to the binding of these types of dyes. Membrane potential dyes. I had to mention these, although not with, well, I can't say not with enthusiasm. I just don't know how they work, or I don't know completely. These types of dyes bind to neurons, and you can see small changes in their emission when a neuron is stimulated. My reason for lack of excitement in that is because the mechanism is not always known. It was hoped that they would be responsive to the electric field across a membrane, but the changes in intensity that are seen are probably not due to the electric field across the membrane, but to changes in partitioning or changes in orientation when a neuron fires. So they work, but not the way it was hoped that they would work, as actually sensing an electric field. Long wavelength probes. These are of great interest, especially for small animal experiments now, because let's just see what they look like first. If you have a flashlight, put a flashlight behind your hand, you see the red light coming through it. And that suggests that if you can have a red fluorescent molecule, you might be able to observe it through the skin. And there are numerous experiments with mice and rats where tumors are grown in the mice and the rats, and the tumors are labeled with these long wavelength dyes, typically by their conjugation with some antibodies, and you can actually see them through the animal skin. Okay, this is only an introduction to chemical sensing probes. These are probes that work by a mechanism of photoinduced electron transfer. So since I know why they work, I like them. And the basic mechanism is that if you have an anthracene or similar polynuclear aromatic with an amino group near it, it's going to be quenched by energy transfer upon excitation of the anthracene. And anything that you can do to remove electron density from the nearby amino group can make it fluorescent. So in this case, in this schematic, we have a phosphate. It's pulling some electron density away from the amino group, and it becomes highly fluorescent. And this mechanism has been used for sensing other types of molecules like this. This same molecule is sensitive to citrate because it also has a bunch of negative charges on it. This photoinduced electron transfer mechanism has been looked at extensively for making probes for glucose, and this has been such an important project for many years now, trying to make glucose sensors that can be used and avoid having to do finger sticks to take blood samples. But all of these sensors work the same way, except they're using a boron group rather than just the nitrogen to remove electron density from the nitrogen. And when a glucose binds to them, the fluorescence goes up in the same way because some of the electron density has been removed from that nitrogen group. I wanted to make this warning again. I made it once before. If you're going to pick a sensor, you want to pick a sensor that is sensing in the ground state and not in the excited state. And one of the most widely used sensors for pH has been HTPS. And the way HTPS works is there's a hydroxyl group here, and when you excite the pyrene moiety, the pKa changes dramatically. So if it was starting at 7.4 in your biological solution, when it's excited, the pKa drops to 1.4, which means it's going to be deprotonated. When it's deprotonated, it's now a different molecule, and it has this long wavelength emission. My problem with using this as a pH sensor is because the excited state reaction seems to will be too dependent on the local environment. So I prefer the Snaffle and Snarfle type probes, which get their sensitivity in the ground state because it's the ionization of the ground state that affects the absorption spectra of these probes, the emission spectrum of the probes, and as I described in the previous lecture, the lifetime, which can be measured at a single phase angle if you so wish. The last types of probes that I'd like to discuss today are the protein probes, which are becoming more popular as genetic modifications become popular. So this is one that is done with the glucose galactose protein from E. coli. And actually, this was done in my lab and other labs. We genetically modified the protein, put it in a sulfhydryl group. The sulfhydryl group was labeled with ANS, which you might remember is a very polarity sensitive molecule. And we found that when glucose binds to the protein, the ANS fluorescence drops. So it's told us the mechanism in which this particular protein sensor worked is by glucose replacing or pushing out the ANS away into a more water environment. A protein sensor can also work in the opposite direction. This is the phosphate binding protein. also from E. coli, genetically modified, to put in I believe it was a coumarin probe, and hoping that it would sense phosphate binding, and that's exactly what happened. In the absence of phosphate, the coumarin fluorescence is very low. And in the presence of phosphate, the coumarin fluorescence increases. So this suggests that the coumarin moiety is shielded from the water by the phosphate group. So this is another type of phosphate, another type of protein sensor. That will be it for the moment. And as I said, there are many types of probes, and I always find this lecture a little difficult to give because I'm just giving a description of a variety of probes in it and how they work. And I hope that helps you select probes for any experiment you might want to do. See you next time.